So now we're going to have Keisha Harrison, who's a PhD candidate at Oregon State University in food science and technology with a focus in microbial ecology and microbiome data analysis. Today, Keisha will be talking about some work she did using R to look at kombucha.
Hello, everyone. Thank you for coming to my talk. My name is Keisha Rose Harrison. I am a fourth-year PhD student at Oregon State University, where I study microbial succession in complex food matrices. Today, I will talk about how the utilization of the R package Phyloseq is extremely helpful in that line of research. So to get started, the food system and microbial system that I study is kombucha. For those of you who aren't too familiar, kombucha is an acidic and live beverage containing small concentrations, often null, of alcohol. It is made by the fermentation of sweetened tea with a mixed consortia of microorganisms known as a symbiotic culture of bacteria and yeast, or a SCOBY. It's usually fermented for 8 to 10 days after being inoculated with the physical phase, commonly referred to as the SCOBY or the pellicle, which consists of osmophilic yeast, acetic acid bacteria, and lactic acid bacteria. As for the experimental design or the story behind the data, I ended up conducting a time course study on an active commercial kombucha fermentation where I collected SCOBY and broth, so physical or liquid, states of the starter culture. After DNA was extracted, the DNA was prepared into libraries that were then pushed forward for a MiSeq high throughput sequencing run at the CGRB at Oregon State University. It's that downstream analysis of this data where we really got to utilize the full robustness of the Phyloseq package in R. By using a series of functions within the Phyloseq package, we're able to take the number of reads assigned to each taxa in a sample and construct a visual representation as well as a statistical understanding of how microbial composition existed in different samples taken at these different time points. So the function merge Phyloseq, for instance, allowed us to kind of coalesce our data tables so that we could look at an overall object that was inclusive of the number of reads, or we'll refer to as our ASV table, the taxonomic assignments, as well as sample information. Transform sample counts gave us the ability to convert these number of reads and look at the relative abundance and understand microbial composition within each sample of each time point. Also, subsetting taxa and subset samples were extremely helpful functions that we were able to utilize to specifically focus on samples that would allow us to address questions within our metadata. We were also able to utilize the Phyloseq package to look at the alpha diversity within our sample population. Here, we're able to look at two groups, our broth samples and our SCOBY, and compare them for species richness and evenness by calculating a Simpson index. Thereafter, we were able to plot, as you can see in this slide here, to really visualize any sort of clustering. Ultimately, with the assistance of another package, we're able to look at linear mixed regression models to evaluate the statistical strength of our comparisons. So piecing our objective and methodology together, the overall goal of this project was to look at the microbial succession of microorganisms within an active kombucha fermentation over the course of 20 days. We're able to focus just in on the primary fermentation, which is a 1 to 7 day process using the subset function within Phyloseq. We were able to process the fungal information as well as the bacterial information by looking at two separate Phyloseq objects. From there on, we're able to visually assess the microbial composition over the course of these 7 days, and we're able to calculate alpha diversity and beta diversity, although beta diversity was not shown, to really look at microbial diversity or microecology with statistical strength placed behind it. From these figures below, we can confirm that there was not an observable or statistically significant relationship between time points or ages of the fermentation on the microbial composition. Phyloseq was extremely helpful in being able to validate the null hypothesis. Thank you, everyone, for listening to this quick talk. If anyone has any questions or comments, feel free to send me a message. My email is on the title slide. Bye.